Good evening, everyone, and welcome to the third series in the Niesler medical series. We are going to be talking about inherited red cell abnormalities tonight, and in particular, sickle cell anemia. This presentation was prepared by our medical scientist, Ashley Welsh, and will be presented by myself and Professor Carola Niesler. Thanks, Carola. If we can have the first slide, please. So to begin with, I will just touch on what red cells and haemoglobin is all about. So as you all know, red blood cells carry oxygen and carbon dioxide through the body and contain the protein haemoglobin that is actually the unit that carries these gases. So hemoglobin is a heterotetramer, which means it has subunits, 4 subunits to its structure. The hemoglobin contains 2 alpha and 2 beta subunits. In each subunit, there is a heme molecule, and it is the heme that binds the oxygen and the carbon dioxide. Now, moving on to gene mutations of hemoglobin, these affect about 7% of the world's population, and they appear in 2 main categories. Your gene mutations that cause an unbalanced production of globin protein subunits, which result in diseases such as thalassemia, or you have the group that produces actual abnormalities in the globin protein subunits, such as sickle cell disease. Next slide, please. Sickle cell disease is a group of inherited red cell disorders where the red cells become very sticky and C-shaped, like the farm implements the sickle. And they're inherited in an autosomal recessive fashion. There are a number of types of sickle cell disease depending on which genes are inherited. The first type is the HBSS, where the inheritance is a sickle cell gene, the S gene, from each parent. This is the most common type of sickle cell disease and is known as sickle cell anemia. The HBSC type, which is an inheritance of the sickle cell gene S from one parent, and an abnormal hemoglobin gene C from another parent. This type of sickle cell disease is usually milder than sickle cell anemia, but has very similar signs and symptoms. Then you have the HBS beta thalassemia, where the inheritance is one sickle cell gene S from one parent and one gene for beta thalassemia. Then, lastly, we have what we call the sickle cell trait. Now, this is denoted as HBAS, where the inheritance is one sickle cell gene from one parent and one normal gene on another parent, the cells in their blood. Next slide, please. So as I said, we'll be concentrating more on sickle cell anemia this evening, and in the previous slide, I mentioned this is the most common type, and it is the most severe type of sickle cell disease. And it is characterized by anemia, vaso-occlusive events, and tissue damage. So now if you look on the right side of the screen at the diagram, these clinical signs and symptoms are produced as a result of these rigid sickle red blood cells, which tend to cause vaso-occlusive events in the microvasculature in the body. And this leads to ischemia and hemolysis of these red blood cells, resulting in, with ischemia, you have resulting in necrosis of body tissues, organ dysfunction, acute pain, and oxidative reperfusion stress. The hemolysis results in fatigue, anemia, cholelithiasis, and endothelial dysfunction. With the hemolysis, you also get free plasma hemoglobin, which acts as a potent nitric oxide scavenger. And nitric oxide is important as reduced nitric oxide levels will lead to inhibition of vascular function of the endothelium and nitric oxide dependent blood flow, which results in visible chest pains in these patients. Next slide, please. So the formation of these sickle cells arises as a result of a point mutation on chromosome 11. There is a point mutation where alanine is swapped by a thymine nucleotide, and the mutation causes a substitution of the protein glutamine to valine in the hemoglobin beta subunit. So upon the hemoglobin releasing its oxygen, a hydrophobic patch of protein becomes exposed. The valine protein that is now that has been substituted can then bind to this patch. As such, a polymer of hemoglobin forms in the red blood cells. As these polymers grow, they distort the red cell's membrane, resulting in its rigid and very sticky shape. Next slide, please. So looking at sickle cell anemia in Africa and South Africa, the most common mutation of sickle cell disease is HBSS in sub-Saharan Africa, as we have said, and the distribution of the mutated HBSS sickle cell anemia gene follows the historical distribution of malaria throughout the continent. It has been shown that those with the sickle cell trait, the ones who only carry one copy of the mutated hemoglobin gene, have resistance to malaria. And now there are various mechanisms that can explain this malarial resistance, some being sickling of the infected red blood cells obviously doesn't allow the parasite to infect the red cell. You have increased splenic phagocytosis, so parasites are killed faster, premature hemolysis and parasite death because of the first two things I've mentioned, and there is weakened sucker adherence of the red blood cell to the site, thus leading to acquired host immunity. However, those patients with sickle cell anemia, sorry, that's incorrect on the slide, it says sickle cell disease, can become infected and are twice as likely to die if they have sickle cell anemia. So they are compromised with malaria. Next slide, please. Initially, sickle cell anemia wasn't really very prevalent in South Africa, but in the 2000s it certainly increased with increased migration from sub-Saharan African countries where it is very prevalent. On the right-hand side, you can see the Democratic Republic of Congo has a 62% prevalence of sickle cell anemia. And as these people immigrated down to South Africa, the amount of cases at the Red Cross Children's Hospital increased significantly, peaking in 2009. Thereafter, we have seen a decrease, and it could be attributed to more available genetic testing. Also, in these African countries, people are tested voluntarily, and they often carry around a little card that will say if they have sickle cell trait or if they have any of the sickle cell diseases. So it really is a thing where couples will show each other their card before deciding to have a child because of the high prevalence of this disease. Also, with better education and, as I said, gene testing and fertility options, the amount of sickle cell has slightly decreased, but nevertheless still prevalent. Next slide, please. So current treatments that are available for sickle cell disease sufferers are blood transfusion and hydroxyurea. So blood transfusions obviously work by providing the individual with normal red blood cells, which reduces the concentration of sickle cells. The pros are that it reduces symptoms of anemia and tissue damage by improving the oxygen carrying capacity of the patient's blood. The cons, however, are alloimmunization to repeated blood transfusion, lifelong transfusions, and blood transmissible infections. The hydroxyurea works by inducing the production of fetal hemoglobin, which inhibits sickling. It is safe to use, it prevents organ damage, protects against cerebrovascular disease, and reduces the need for hospitalization and blood transfusion. However, access to the drug, patients don't always have availability, and some are not really willing to use the drug. It is not always effective, and it is unable to help treat patients that have a progressive vasculopathy. Next slide, please. The current cure for sickle cell disease is haemopoietic stem cell transplantation, and the stem cells can be retrieved either from the peripheral blood or bone marrow of a donor or umbilical cord blood. With umbilical cord blood, it must be noted that the child cannot use their own umbilical cord blood, as it will contain gene mutations for the hemoglobin. So the child would either have to use cord blood from a donor or from a matching sibling. So how it works, the patient, the donor will give the peripheral blood or bone marrow stem cells, or cord blood will be collected. These stem cells will undergo processing and cryopreservation. In the meantime, the patient will undergo chemotherapy to eliminate the dysfunctional bone marrow, and then will be infused with these stem cells that will regenerate a new bone marrow for the patient. And stem cell transplantation is being done in South Africa to treat children affected with sickle cell disease. So now I will hand over to Professor Carola Niesler, who will take you through a couple of interesting articles on the use of cord blood in the treatment of sickle cell disease and gene therapy. Thank you very much. Over to you, Carola.
Thank you, Yvonne. Good evening. So, as mentioned earlier, haematopoietic stem cell transplantation is currently the only cure for sickle cell disease. And it has an excellent cure rate of exceeding 90%, but the availability of suitable adult donors can be an issue. And so transplantation of umbilical cord blood stem cells from a related donor, which results in survival rates that are the same or even better when
Carola, are you there?
Yes. Yes. Hello.
I can hear you, Carola.
All right. So you could hear me. Sorry, Yvonne, I think, could not hear me. So I'll just carry on. Okay.
Yes, I think it's fine.
Okay, perfect. Just to pick up where I left off, transplantation of cord blood stem cells from a related donor is an option, an alternative option to hematopoietic stem cell transplantation. And the results using cord blood stem cells are equivalent or better than when using hematopoietic stem cells from an adult donor. And additionally, there is a lower probability of acute or chronic graft versus host disease using the cord blood stem cell option. So our first paper tonight that we're going to be looking at focuses on the use of cord blood stem cells and is entitled Family Cord Blood Banking for Sickle Cell Disease, a 20-year experience in 2 dedicated public cord blood banks. So in this example of directed cord blood banking, cord blood units were collected between 1995 and 2014. Participants were pregnant women who had a child homozygous for sickle cell disease, and 338 cord blood units were ultimately banked and reserved for 302 families. So 209 in cord blood bank 1 on the left and 129 in cord blood bank number 2. So although the majority of units banked were from families that involved a single time, there were 32 mothers recruited more than once, these banking up to 4 cord blood units per family, as seen highlighted in red in the boxes at the bottom of the slide. So cord blood units were transported and processed within 24 hours of collection, and only once the decision to proceed with transplantation had been made was HLA typing carried out. And then the standard cell counts and cell viabilities, total nucleated cells, CD34 numbers, sterility testing, etc., all those functional assays were then also carried out. Over the 20-year period, a total of 28 out of 338 cord blood units were transplanted. So this is a low utilization rate, representing only 8% of the banked units. The median age of the recipients at transplantation was 6.8 years. So on this table, you can see on the right-hand side, boxed in blue, are the total cord blood units. So there are 28 in total. And then the 2 columns to the left of that, which are outlined in red, show cord blood units infused alone or cord blood units infused with bone marrow. The difference between these two is that the column in the middle was for the case where the 5 cord blood units with a total nuclear count of less than 20 million per kilogram were available. So in this case, they were combined with bone marrow from the same donor, and the results indicated in that column. The remaining 23 on the left didn't require this addition of the bone marrow. So across all patients, then all the 28 patients, the average collected cell dose was 40 million total nucleated cells per kilogram, and of those 160,000 were CD34 positive. The average infused cell dose was 31 million total nucleated cells per kilogram, with 140,000 CD34 plus cells per kilogram. And the average recipient body weight was 22 kilograms. So 27 out of the 28 transplant patients are alive and free of sickle cell disease symptoms. And this includes follow-ups ranging from 4 months to 14 years. 5 patients of those who received a single cord blood transplant did experience acute graft versus host disease, and of these 4 required steroid treatment. 1 recipient of the combined cord blood and bone marrow transplant did develop limited liver chronic graft versus host disease, which was the only chronic GVHD event reported for the entire cohort. The 28th patient did have primary graft failure. This patient is alive, though anemic, but has had no vaso-occlusive events, so that's a good sign. The patient was transplanted in 1996 with an adequate cell dose, but had primary graft failure and unfortunately failed to engraft after second bone marrow transplant. So the conclusion from this paper was that an adequate volume and total nucleated dose is important, total nucleated cell dose is important for a successful transplantation to treat sickle cell disease. However, where you encounter low volume or low total nucleated cell dose collections, you can use bone marrow to substitute, and therefore low cell numbers should not be a limitation for the use of these cord blood units, especially if there is a chance to increase the cell number by using the same sibling donor. So possibly this could help improve on the low utilization rates, 8% in this particular study. Of interest is that these low utilizations of banked sibling cord blood units for hematological disorders have actually been reported by a number of other groups. So in our next paper, entitled Highly Efficient Editing of the Beta Globin Gene in Patient-Derived Haematopoietic Stem and Progenitor Cells to Treat Sickle Cell Disease, here we're going to take a slightly different track. So here the authors use the CRISPR-Cas9 system to correct the sickle cell mutation in the affected gene. And the study uses bone marrow-derived and peripheral blood stem cells, human bone marrow-derived and peripheral blood stem cells, and analyzes their ability to differentiate it into red blood cells following the gene correction, and also their ability to engraft in vivo in a mouse model. So before we look at the results and the study, let's just take a very quick look at what is CRISPR and what is the Cas9, CRISPR-Cas9 system. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And these repeats are always found next to CRISPR-associated genes, and these genes code for endonucleases, and these are the enzymes that cut DNA. And these genes are known as the Cas genes and encode the Cas protein, the endonuclease. The palindromic repeats cause the DNA to form a hairpin structure, and this is very important because it ensures that the Cas9 endonuclease cuts where it's supposed to. So in this way, the CRISPR system enables precise cutting and gene editing. This can be addition, can be removal, or alteration of genetic material. Importantly, previous methods had far less control over where and what was actually being edited. So the CRISPR-Cas9 is often referred to as a molecular scissor approach. So the 2 main components, the Cas9 being the endonuclease, and then the second component that's really important is the guide RNA, which is the target or the guide sequence and is typically 20 base pairs long. So as shown in the schematic on the right, the CRISPR gene editing would work as follows. You have your target sequence, and a guide RNA sequence will bind to that target gene sequence. The Cas enzyme will then bind to the guide RNA and will cut the double-stranded DNA. And this DNA damage is then repaired using innate cellular repair systems. And the approach can then be used to incorporate a desired DNA sequence into the molecular cells, into the cell's genome. So in the study, they used this CRISPR-Cas9 system and a short single-stranded oligonucleotide template to correct the mutation in the beta globin gene in stem cells. they made use of homology-directed repair, and this uses a double-stranded DNA donor that has homology to the adjacent sequences surrounding the lesion to incorporate the new DNA fragments. As part of the optimization, they designed various CRISPR guide RNAs that target the first exon of the beta globin gene, with varying proximity to the sickle cell disease point mutation that Yvonne referred to. So the R66, and if you have a look on the left-hand side, you'll see a number of sequences, and right at the bottom you can see in red the A, which is the point mutation change from the T. So that R66, is shown second from the bottom on the left, was the sequence selected as the guide RNA that worked best. It caused a double-stranded break, one base pair away from the sickle mutation. Then they selected the best single-stranded oligonucleotide template, and on the right you can see a graph showing the percentage homologous direct repair, and then on the bottom on the x-axis you can see the different single-stranded oligonucleotide templates, and underlined in red was the best one for this particular approach, giving the highest frequency of homology-directed repair. So they first investigated then the gene editing success of the CRISPR approach in vitro. And what they did here is they used the optimized Cas9 and then the optimized guide RNA, which had the optimized corrected sequence. And they used peripheral blood and bone marrow-derived haematopoietic stem cells from sickle cell disease patients. And after 21 days of differentiation of the gene-edited sickle cell stem cells, they could then calculate the percentage beta hemoglobin in these cells. Right? So following the gene editing, they then wanted to see whether that gene editing process had improved the percentage of beta hemoglobin in those particular cells. And indeed it had, in both the peripheral blood stem cells and the bone marrow stem cells. When they were differentiated in vitro, the levels of hemoglobin, beta hemoglobin correction had increased 37% and 33% in the peripheral and bone marrow stem cells, respectively. So then, following on from those in vitro studies, the group carried out some in vivo studies in mice, and they particularly used the non-obese diabetic SCID gamma mouse model. So those are the NSG mice. So these mice are immunodeficient. They're unlikely to cause alloimmunization, so that's the induction of the immunity in response to the foreign antigens, and there's a complication of repeated blood transfusions. They analyzed engraftment, so where the blood-forming cells start to grow and make the new healthy cells, and they really wanted to determine whether the gene-edited cells retain the ability to engraft. So, and to do this, the CD34 positive bone marrow stem cells were treated with the Cas9 guide RNA and the selected single-stranded oligonucleotide template. They were then injected intravenously into the NSG mice, and 16 weeks after transplantation, the bone marrow was harvested and analyzed for engraftment rates. So overall, the result was that both bone marrow-derived and haematopoietic and peripheral blood stem cells that were gene-edited were well tolerated by the NSG mice. And shown here are the results from the bone marrow haematopoietic stem cells at 16 weeks after transplantation, showing variable but generally good engraftment. So what you can see on this graph is the engraftment on the y-axis. So the higher up you go, the better the engraftment. And on the x-axis, you've got the HDR, which is the homologous direct repair on the left. And then you also have the addition of the non-homologous end joining. And I'll explain very briefly just now why that was added. So the different colors there, the donor 2 RNP would be the donor cells treated with the guide RNA and the Cas9. But no template, right? No oligonucleotide template. And I remember that the HDR method, the homologous directed repair method, needs a template. Then the yellow and the green are two donor populations which have been treated with the guide RNA, the Cas9, as well as this optimized template. And then the blue, dark blue, is the donor cell with simply the template, but not the Cas9 or the guide RNA. And then the light blue is the untreated with just the donor cells. And so you really would like to see the green and the yellow for the HDR being as high as possible. And you do see that from the green, there's quite a lot of scatter, and the yellow is higher than any of the others as well. So that indicated that engraftment was, all the variable was generally good. When you look at the non-homologous end joining, the NHEJ, now the NHEJ break ends, they can be ligated without a homologous template, so they don't need that single-stranded ODN that's shown for the yellow and the green. So whereas the HDR, so the homologous directed repair, requires a template to guide the repair. And the NHEJ is a very efficient repair mechanism. It's very active, but it's very susceptible to mutations and errors. And so the HDR, the homologous directed repair mechanism, is actually preferred in this case. But what you can see is that the red dots, which were basically your Cas9 and your guide RNA without the template, they suddenly show good engraftment as well. And that's the difference between the homologous and the non-homologous end joining section of this particular graph, which would make sense given that the second method doesn't need a template. So in summary, the only cure for sickle cell anemia is hematopoietic stem cell transplantation. However, this approach is limited due to donor matching. And so autologous hematopoietic stem cells that have been genetically edited to exclude the sickle cell mutation may offer a solution to the limits of allogeneic hematopoietic stem cell transplantation. And over the last 20 to 25 years, we've seen huge strides in gene editing techniques. The CRISPR-Cas9 being probably one of the most recent and breakthroughs in this particular technology. And so hopefully we'll see some good results coming from bigger clinical trials soon. Thanks very much.
Thank you so much, Carola and Yvonne. That was great. I do have one question here, and it's for you, Yvonne. Can you just confirm if you can hear me? Yvonne?
Yeah,
oh, there we go. Okay.
Yes, here I am.
What is the difference between beta thalassemia major and beta thalassemia minor?
So it's all to do with the subunits of the haemoglobin, and they are produced in different numbers. You also get different, beta thalassemia major is more severe than the minor one, and their life expectancy is also very different. So they can be two different diseases.
Great, thank you so much, Yvonne. I don't have any more questions, but if anyone does have questions, they can add them to the question box. I'm just going to wait a few minutes to see if anyone has some questions, and then we can take it from there.
Hello, can you hear me?
Yes. Yeah, I think the signal is a bit bad, but I don't have any more questions, Yvonne. So okay, so because we don't have any more questions, I think that is the end of our webinar. Thank you so much for everyone who attended, and please keep safe in this very terrible third wave.
Thank you, Ali.
Thank you, Ali.